When we talk about the biggest challenges in modern medicine, treating diseases of the brain, like Alzheimer's, is right at the top of the list. It's an organ that's protected by this incredible defense system. But what if we could actually turn one of the disease's biggest risk factors into a weapon against it? Well, a recent study did exactly that, and it gives us this fascinating peek into the future of gene therapy. To really get why this is so important, you just have to look at the numbers. By 2050, we're expecting the number of Alzheimer's cases in the US alone to just skyrocket to almost 14 million people. I mean, that's not just a health crisis, right? That's a societal one, and it makes finding new ways to fight this disease more urgent than ever. Okay, so at the very heart of this whole story is what you could call a genetic lottery. See, there's this gene called APOE, and it comes in different versions. If you inherit the APOE4 version, your risk of developing Alzheimer's goes way up. But, and this is the cool part, if you get the APOE2 version, it's actually protective. It cuts your risk by about half. It's like a classic case of a good twin and a bad twin written right into our DNA. And that leads to this, just brilliant idea at the core of the research we're looking at. If APOE4 is the problem and APOE2 is a potential solution, what if we could deliver the protective APOE2 gene directly into the brains of people who have the high-risk gene? Could we basically use gene therapy to stack the deck back in the patient's favor? So how do you actually do that? Well, you need a delivery vehicle. For this, scientists use something called an AAV vector. The best way to think about it is like a microscopic delivery truck. This AAV is a virus that's been totally hollowed out, made completely harmless. Its new job? To carry its precious cargo, the good APOE2 gene, and its destination is the cells of the brain. But here's the catch. Even with the perfect delivery truck, you still have to get past the fortress walls. The brain and our central nervous system are incredibly well protected, which makes it super difficult for most drugs and therapies to get inside and do what they need to do. Honestly, this is the single biggest hurdle for treating most neurological diseases. And that is the exact question this study set out to answer. See, it's not enough to just get the therapy to one tiny little spot. For a disease like Alzheimer's, which affects huge regions of the brain, you need it to go everywhere. So the quest was on to find the perfect delivery route, one that was effective but also as safe and non-invasive as possible. So the researchers set up what was pretty much a three-way race to the brain, and they tested it in non-human primates. So in lane 1, direct injection, going right into the brain tissue itself. In lane 2, ventricular delivery, which goes into the fluid-filled spaces deep inside the brain. And in lane 3, cisternal delivery, into a reservoir of spinal fluid right at the base of the skull. So how'd our first racer do? Well, direct injection kind of worked, but only locally. It delivered the gene to the specific spot it was injected into, but it completely failed to spread out. It didn't get that all-important widespread distribution. So a decent start, but definitely not the home run they were hoping for. But the other two methods, wow, that was a totally different story. By putting the AAV vector into the cerebral spinal fluid, that's the liquid that basically bathes the entire brain and spinal cord, both the ventricular and the cisternal routes were a massive success. The protective gene got distributed far and wide, all throughout the central nervous system. It's just a brilliant example of using the brain's own natural plumbing system. Okay, so now we have two winners, but are they created equal? Not even close. Yeah, both methods got the job done, but the key difference here is how invasive they are. Intraventricular delivery, that means brain surgery, drilling a hole in the skull. Intracisternal delivery, on the other hand, is a much simpler procedure, almost like getting a spinal tap. And that gives us our clear winner. When you have two effective options, you're always going to go with the one that is dramatically less invasive and safer for the patient. So the optimal strategy found by the study was, without a doubt, intracisternal delivery. So let's just recap the victory lap here. The intracisternal route won because it checked every single box. It gave them the widespread gene expression they needed for the whole brain. It was by far the least invasive option, so you avoid all the risks of major surgery. And on top of all that, it showed an excellent safety profile. Efficacy, safety, and simplicity. That is the trifecta. And hey, you don't have to take my word for it. The study's own conclusion says it perfectly. They found that intracisternal delivery was the optimal strategy for getting this therapy safely and effectively where it needed to go. That's really the headline from this whole paper. Now, we have to be really clear about something here. This study is not a cure for Alzheimer's. Not yet. What this was a preclinical study that was laser-focused on one specific, but absolutely vital piece of the puzzle. How do you deliver the therapy?
So if it's not a cure, why does this matter so much? Well, first, it solves a massive logistics problem for this specific gene therapy, which brings it one giant step closer to being tested in humans. And maybe even bigger than that, it helps pave the way for a future where treating these complex brain diseases might not require invasive brain surgery, but something much, much simpler.
And that just leaves us with this incredible, powerful thought. This research opens the door to a future that honestly we could barely even imagine a decade ago. A future where maybe, just maybe, a single, simple injection could deliver a protective gene that helps prevent one of the world's most devastating diseases. The journey is still long, for sure, but this study has just charted a very, very promising new path.